Okay, we did probiotic diversity in yogurt. I'm Hannah, and then my group mates are Jasmine and Becca.
E. Okay,
Okay.
so for a little introduction, our research question was, is there a difference in probiotic diversity between Chobani's non-fat plain Greek yogurt, Yoplait, and Go-Gurt? So for background information, we found studies done on female mice, including dairy products from cow yogurt, and that they increase probiotic diversity in gut microbiota. These probiotics, including the one listed, helps decrease weight gain in the female mice. Another study we found was on the benefits of probiotic use for humans, concluded the use of probiotics to support gut function during antibiotic use or to reduce the risk of respiratory tract infections in healthy individuals. That led us to our hypothesis and prediction. Our hypothesis was that Chobani's plain non-fat Greek yogurt will have a greater diversity of probiotics than Yoplait and Go-Gurt because additional cultures are added to the Greek yogurt during fermentation. Our prediction is if Greek yogurt is considered healthier, then Chobani will contain a greater diversity of probiotics over Yoplait and Go-Gurt.
To test this, we got samples of yogurt, like Becca said, Chobani, Go-Gurt, and Yoplait, and we ferment or grew them in anaerobic and aerobic cultures. Then we gram stained each sample and created streak plates. Our secondary tests were the catalase tests. And we, for all the isolates, and then we performed different tests to deduce what exact microbes we had. And then we observed the test results and compared them to biochemical profiles and found our microbial species.
So these are our results. We found two Chobani isolates, three Gogurt isolates, and two Yoplait isolates. And we measured their characteristics. So if they were aerobic or anaerobic, they were all gram positive, and then we documented the cell shape. We also wanted to look at enzymatic activity with catalase and oxidase and fermentation of glucose and mannitol, as well as lysis of red blood cells. Our findings were that all of our isolates were gram positive. All of the catalase and mannitol tests were negative. Glucose fermentation in the acid was common in most tested aerobic and anaerobic isolates, and they had mixed respiratory capabilities. So there were some comparable differences there were comparable groups despite the brand differences.
Based on our data, our hypothesis was not supported because we could not distinguish between the different species of gram-positive rods. We needed to do further research to distinguish between the varying species in the yogurts. We were unable to say if Greek yogurt is really more diverse because we could not fully distinguish between the microbe varying species. Here's some research on microbial contamination in yogurt, and if companies follow good manufacturing practices, then we would not find contaminants. So it's likely that the yogurts we looked at did follow these because we were not able to find E. coli or salmonella.
Some of our limitations included that it was difficult to isolate all the colonies. Initial colonies on the streak plates included mixed cultures, and even when we did a second streak plate, it was still hard to try and isolate the two. This is because Streptococcus and Lactobacillus have a symbiotic relationship in fermentation, so they feed off of each other. So that's why we couldn't really isolate the two from each other. Another limitation is that majority of live cultures in yogurt are gram-positive rods. So all of the extra cultures that are added to the Chobani Greek yogurt, they are all gram-positive rods. So we couldn't really distinguish between species of if it was just from fermentation or if it was additional cultures added.
So in conclusion, our results were congruent with what we expected. We found Streptococcus and Lactobacillus in all of the yogurts. However, they did not differ between yogurt brands. The diversity findings were measured via Berkey's Manual of Determinative Bacteriology.
Some possible further research is studies of isolate cultures to identify the individual species. And then another research is the uniqueness of how cultures grow at different temperatures. So possible research would be testing variations in temperature on the presence of Streptococcus. And these are our literature cited. Thank you.